So we are going to deal with the continuation of other development parts of the neural tube. So a third was forming as the spinal cord. Then the cranial two-thirds is going to form as the brain. So the brain takes a longer length or percentage of the developing neural tube. So the brain also starts to develop after the closure of the cranial neural pore, which we say closes on the 25th day of intrauterine life. So after closure of the cranial neural pore, we start to form what we call vesicles, brain vesicles. So the brain vesicles are like thickenings in the area which are going to form for us the future brain. The brain vesicles, there are three vesicles which are formed. So starting from the connection of the spinal cord, we have what we call the rhombencephalon. That is the part which will form for us the hindbrain. So rhombencephalon. Then up next is the metencephalon, which is going to form the midbrain. So we call it the midbrain vesicle. Then we have the prosencephalon. The prosencephalon is the forebrain vesicles. The forebrain vesicles, that is the part which is going to form for us the future forebrain. Now, as development continues, the prosencephalon and the rhombencephalon undergoes further division. The prosencephalon, which is the forebrain vesicle, further splits into two components, which we call the telencephalon and also the diencephalon. Then the midbrain vesicle, that is metencephalon, does not split, it remains the same. Then the rhombencephalon always splits also to form other two components. The most downer one we call the myelencephalon, which is a part which is straight away a continuation of the spinal cord. Then next to it is the metencephalon. So now, in the process of development, if we come from the spinal cord connects to the part of the brain vesicle, which is the rhombencephalon, but specifically the lower part of the rhombencephalon, which is the myelencephalon. So in the development of the brain stem, that's the hindbrain, it still follows the same makeup like that of the spinal cord, where we have a central opening, that is a hole. Then we are having the three layers where we shall have the aggregation of cells, which we shall call them the Alar plates and the Basal plates. But to note very importantly is that in the brain vesicular development, starting from the hindbrain vesicles, there is a bit of eversion in that the Alar plates, instead of being exactly on the dorsal side, they be on the lateral side, like they be lateral to the basal plates, such that if you're drawing, if you're having a line which is running in the dorsoventral position, it does not exactly cut through both the ventral and the Alar plates. So due to eversion of the brain vesicles, you find the Alar plates are more of lateral. And this one, you shall also appreciate it that in the brain, actually the gray matter is outside, meanwhile the white matter is inside, as reverse to that of the spinal cord. Reason being, there is a process of eversion that occurs. So, in the development in the hindbrain, we have the Alar plates and the basal plates. The cells or the neurons in the Alar and basal plates further split into three different groups. In the Alar plates, we are going to have three different groups, and also the basal plates, we are going to have three different groups. Now, since we are saying the Alar plates shift and be more lateral compared to those in the spinal cord. We are going to have three. In the hyalar plates, we are going to have three different groups, starting from the medial, intermediate, and lateral. The same thing in the basal plates, we are going to have the medial, lateral, and intermediate groups of cells arising from the basal plates. Now, get to know that the cells which are in the hyalar plates, they always form cells which are always receiving information. That is to say, the afferent. Meanwhile, cells of the basal plates, they always form outflow, what we call efferent. So now, we are going to find efferent, that is the one sending information, which are going to a special group of organs. We're going to have those which will go to mainly skeletal muscles of a certain region, and we have those which are going to the internal organ, what you call the viscera. Then the same thing will happen to the afferent, the one which are bringing information. Now, starting with cells of the basal plates. The cells of the basal plates, we are going to find it splitting into three different groups. So if we are to arrange from medial to lateral, starting with cells from the basal plate, we have the somatic efferent, we have the special visceral efferent or special efferent. Then we have the third group, lateral, which is a general somatic efferent. Now, when it starts on the side of the hyalar plates, for them they have the arrangement different. So we realize here somatic was the most medial. But on the side of the hyalar plates, the visceral, so the somatic afferent is going to be the most lateral. Then special afferent is going to be the next. Then the most medial in the hyalar groups will be the general afferent. So like that is a bit of the difference in arrangement. So the general afferent is the most lateral on the basal. Then the general afferent is the most medial on the side of the hyalar plates. Now, the next thing we are going to do is to know which nuclei will be a component of the somatic afferent or the somatic efferent, then special visceral afferent or special visceral efferent. So that is the next thing we are going to do. Now, before that, we have talked of the forebrain vesicle, that is prosencephalon. We have the midbrain vesicle, which is the mesencephalon, and we have the rhombencephalon. Remember, all these are having cavities which is forming them. So, starting from down, we said the cavity of the spinal cord gives us the central canal. Now, when it comes to the rhombencephalon, that is the part which is the most connected to the spinal cord. The cavity of the rhombencephalon gives us what we call the fourth ventricle. So, due to eversion, where we are saying that the hyalar plates shift to go more lateral, we are going to have what we call the roof and floor. Now, due to eversion of the hyalar plates, the roof is going to remain very thin. So the thin layer of this roof is going to be consisting of mostly the inner, just part of the inner layer of this developing neurotube, which was consisting of the neuroepithelial cells. Now these neuroepithelial cells, for them, they are going to aggregate and differentiate lastly to give us the ependymal cells. Now also on the roof, you're going to have vascular migration. The vessels come from the mesodermal layer of the germ disc, or the three germ layers, the mesoderm that give rise to the vessels. So when the vessels come and lie on top of the ependymal cells on the roof of this developing fourth ventricle, the two layers together, they unite to form what we call the stellar choroidea. Now, on that roof, which is forming the roof of the fourth ventricle, you're going to have some bit of absorption that is going to happen on the roof. The absorption occurs in 3 places, 1 in the midline, then 2 on either sides of the midline. So those foramina will be on the surface, on the roof of the fourth ventricle. In the midline, it will be called foramen of Magendie. Then on either side, we are going to call them foramina of Lasker. So you're going to have Lasker foramina on either side, then foramen of Magendie. This one form a communication between the cavity of the fourth of the rhombencephalon, which is the fourth ventricle, and the outside, which will be the another space outside, which is a subarachnoid space. So it will form a point of flow of the CSF, that is spinal. So now we shall summarize the different groups of nuclei at once, so that before all the brain stem, that's starting from the rhombencephalon, where we have the myelencephalon, metencephalon, and also the Hindbrain, where we have the metencephalon. So we had talked of the Alar plate giving us 3 groups of cells aggregating. Then also the basal plate giving us 3. So the basal plate, they form the efferent. So we have what we call the general-somatic efferent. So for general-somatic efferent, for them they mainly go to groups of skeletal group, a special group of muscles. So the general-somatic efferent, the one in the myelencephalon, is going to be the nuclei of cranial nerve 12, that is hypoglossal nerve, that is going to go to muscles of the tongue. Then, for the case of in the metencephalon, we are going to find the nuclei of cranial nerve 3, 4, and then 6. Now, get to know that the cranial nerve 3 and 4, in a normal person, in a mature person, they come from the mesencephalon. So now, what happens is that there is always migration that happens. Most of the cells will originate from the metencephalon, but finally they will migrate cranially and settle in the metencephalon. That's why the cranial nerve 4 and 3, their nuclei, now in a mature person, is originating from the midbrain, that is the mesencephalon. Then we have what we call the special visceral efferent. The special visceral efferent, this one, they go also to muscles which are derived from the pharyngeal arches. That's what we call the special visceral efferent. So they go to muscles derived from the pharyngeal arches. So on the myelencephalon, they give nuclei of cranial nerve 9 and 10, that is glossopharyngeal, then vagus. Meanwhile, from the metencephalon, they are for nuclei of cranial nerve 5, then cranial nerve 7. Now, still on that way, you know that cranial nerve 5, its nuclei is mainly from the pons. That's where we have the metencephalon. Then we have the general visceral efferent. General visceral efferent, they always have 2 groups of nuclei, one in the one from the myelencephalon and the one from the metencephalon. On the myelencephalon, they mainly form nuclei of muscles of the airway. So, not muscles, like they form outflow to the internal organs of the airway, and they are mainly for cranial nerve 9 and cranial nerve 10. That is from the myelencephalon. Then from the metencephalon, for them they go to cranial nerve, they go to muscles of the sphincter of the pupil, and that is going to be cranial nerve 3. But remember, we say cranial nerve 3 comes from the mesencephalon. In a normal person, there's always migration that happens. That is on the basal plate. On the halar plate, they form the inflow, what we call the afferent. Now, for them, the afferent, we always have what we call the general visceral afferent, which is the medial component. On the basal plate, the general visceral efferent was the most lateral component. In this case, is the most medial component. So, for the general visceral efferent, they mainly go to, they are derived from nuclei of cranial nerve 10. That is a general visceral afferent, which brings information from the internal organs, mainly the GIT tract. Then we have the special efferent. The special efferent, they are mainly coming from taste organs, maybe the taste buds, the pharynx, and where we have those taste receptors. So for them, they are mainly, we have cranial nerve as 7, that is facial nerve from the metencephalon. Then we have, from the metencephalon. Then we have cranial nerve and 9 from the myelencephalon. Then also, the special visceral, the special efferent also can bring sensation from other areas of the head and neck, where we also have cranial nerve cranial nerve 8, which is coming from the cochlea. They also have special sensation coming from the viscera, that is the cochlea, and the vestibular apparatus, that is cranial nerve 8, which is coming from the metencephalon, which is the pons to be. Then we have what we call the general visceral efferent. So general visceral efferent, it means they bring other general sensation, touch, temperature, proprioception, and vibration from head and neck region as well as the pharynx. So in that case, we have cranial nerve 5, we have cranial nerve 6, we have cranial nerve 9. This one, they bring sensation from other areas. Even cranial nerve 7, they bring other information. So cranial nerve 9, it comes from the myelencephalon. Cranial nerve 5 and 7, they are part of the metencephalon. So this one summarizes the development of those nuclei. Now in the myelencephalon, also the cells of the halar plates, they give rise to group of cells which will aggregate and form for us the olivary nuclei, whose function will be described in details when we are dealing with structure and function of the spinal cord and brain. Now, on the metencephalon, that is where we are having formation of the pons and the cerebellum. The myelencephalon mainly forms the medulla oblongata with its nuclei. The metencephalon forms for us the pons and the cerebellum. So, in the process of development in the metencephalon, we have, on the ventral side, we have the cells of the halar basal plate, which are going to give us the nuclei, which we have just summarized them previously. Then we have on the halar plate, we're going to have development of what we call the rhombic lips. The rhombic lips are going to develop on either side of the halar plate, and they start approaching each other on the midline, mimicking the process of what we are forming the neural tube. These rhombic lips, when they join together, they develop later to form the cerebellum. In that development, in a starting around the 12th week of intrauterine life, that's when we have the cerebellum fully formed, and we have that ridge that develop on the dorsal side to form for us the vermis of the cerebellum. After forming the vermis on this either side where we're having the rhombing lips, we're going to have contours developing, fissures developing. These fissures are going to separate as the different parts of the cerebellar hemisphere, which will be described in the structure of the cerebellum in details. Then anteriorly, so ventrally, where we are developing the pons, other cells of the basal plate will later give rise to what we call the pontine nuclei. So the pontine nuclei are part of the gray matter. Parts of the white matter will be for passage of structures from the higher brain centers to the lower brain centers, when you're talking about of the descending tracts and ascending tracts. The nuclei, the pontine nuclei, they also serve as a point of connection between the cerebrum and the cerebellum. The tracts from the cerebellum comes and pass via the white matter of the pons, and they synapse with the pontine nuclei, and then they proceed and synapse with other cells which develop in the cerebellum. So the metencephalon and the myelencephalon, their cavity will form for us the fourth ventricle, as we had said